Dr. Vijay Chandru on this topic of from human engineered intelligence to re-engineered biology, a journey. So it's so Vijay Chandru got his PhD from MIT on the area of mathematics in its application to decision sciences, and then he worked in the US as a professor at UPenn, was it? At Purdue, sorry, at Purdue, on a variety of things, including computational biology and optimization and other subjects. And then he came back he joined the Indian Institute of Science in the early 90s, where I had also joined a little bit before as a professor in the computer science department. And subsequently, he made a very unusual move of starting multiple companies. So he started Strand Genomics. He was also involved in the development of the Simputer. And he and his colleagues really set the framework of how IISc should handle innovation and startups by faculty members, and they helped develop all the policies that later on many of us could take advantage of, not only in IISc, but many other institutions in the country then have followed those policies. So he continued on at Strand Genomics, which then was bought over by various people, and more recently he has been involved in another startup called CRISPR-Bits. So I think for all of us, and especially for the students who actually, there are a few of them over here, it'll be very interesting to hear about this journey, because for students doing science in India, the normal path is to go on to do a PhD. At IISERs, very often the PhD will be outside the country, and then they either stay on outside or try to come back to India for an academic position. And there are really very limited numbers of academic positions, and the funding position the past few years has also not been particularly encouraging. So, and there are a lot of other opportunities that are available to people doing science, and Vijay Chandru is one of the best people to tell us what they are. So of course, besides all his these other accomplishments, he has various awards, including the Hari Om Ashram Award and an award from the World Economic Forum as a technology pioneer. So once again, it's a great pleasure to have you here, Vijay, and we look forward to your talk.
Thank you very much, Raghavan. I think, first of all, I would like to thank the Academy for this honor of giving the public lecture. I've attended a few, and I became a fellow of the Academy in 1996, and I, of course, was reminiscing the early days of Resonance when we were all co-editors, and Professor Mukunda had started the Resonance magazine, and so I think I had checked with Raghavan whether I should give a scientific talk or talk about the journey, and he thought the journey would be interesting. So forgive me, there's not enough math and biology in the talk, but sort of enough to give you a sense of how the journey went. So, well, first of all, I think this theme of human engineered intelligence to re-engineered biology I did write and guest editorial in Current Science. This was right in the middle of the first wave of the pandemic. And so in many ways, if you want a short 2-page description of what this talk is about, it's there online, and you can go take a look. As Raghavan mentioned, I was a professor of Purdue, and by the way, I didn't bring it up when we had the fellows meeting, but Professor Ramdas was, of course, one of the wonderful senior faculty there who was very welcoming of young faculty like me. And of course, we knew him well and had many occasions to dine at their place and vice versa. One of my visits back to Purdue in the mid-90s, just before I joined the academy, I think a colleague there said, you know, there's this new magazine called The Wired, and the editor-in-chief and the founding editor, Kevin Kelly, has written this unusual book. And he said, you should read it, because I had been telling him that there were some folks at IISc who were trying to convince me to learn a little biology. So he said, you'll enjoy this book. And sure enough, you know, I think this book really lit the light. And if Raghavan recalls, I think we organized a JNCSR discussion meeting that year in 2005. Professor Preeti Shankar and I organized it, and where we brought biologists and computational scientists together to sort of kick off this interaction between computer science and biology. And more about Raghavan's influence as we go through the talk, right? So, you know, this is a fun book to read, by the way. It's you know, it's still available online and in Kindle and at Amazon and so forth. Right? Now, I went back and did a little research on my academic lineage, and it turns out that I have 2 Nobel laureates. One of whom actually taught me, Bob Solow, because my minor field was in mathematical economics, and he was teaching at MIT then. And but most importantly, my lineage goes to Leibniz. And for many of us in computer science, Leibniz is really the founder, right? One also thinks of Leibniz as the founder of calculus, but in many ways he was the founder of symbolic reasoning, formal languages, theoretical computer science, if you really sort of take it back, really all draws from Leibniz. And here's a translation by Russell of some of what Leibniz said about, you know, the 2 sides of the coin, one being characteristica universalis, which is, you know, formal languages, and the other side of the coin being calculus ratio connotor, so computation. And in many ways, we thought of this as 2 different cultures coming together. You know, computation and symbolic reasoning somehow belong to different cultures, right? But they're perfectly suitable for solving problems in one domain or the other. And that's why we called it eating sauerkraut with chopsticks, right? See, you know, chopsticks are perfect tools to eat sauerkraut with, as you would eat noodles with, but they come from different cultures, right? So, and then when I became a fellow of the Academy, of course, Rodham Narasimha was the president, and I had the pleasure of getting to know some of his work on the history of science, and he was also a historian of sorts, of course, a serious historian. He's published, and he had this notion of computational positivism, which he felt came from sort of the cultural tradition of classical science in India, and how from observation and computation you get to knowledge, right? That's basically AI, isn't it? I mean, data and computation, and you get to insights, right? So, this was very interesting, and we spent many hours. We traveled overseas and had many conversations about this. So, in many ways, he was my mentor at IISc. And at NIAS, when he was heading the National Institute of Advanced Studies. I was on the faculty there. So here's a little bit of math. I think the math that I was working on was really, a very classical variable elimination to solve different types of problems that could be posed either symbolically in logic, in which case you would use the principle of resolution, which involves, again, elimination of variables. It could be systems of linear inequalities, where again, through basic arithmetic cancellation of positive and negative coefficients, you eliminate variables. And this was Fourier's method for solving systems of linear inequalities, which of course is linear programming, which is a huge problem in engineering systems. And in fact, in those days, when I was doing my PhD, about 25% of the CPU usage of all computers in the world was on solving linear programming problems, right? So this was actually a very important problem. And then, just like you do Gaussian elimination over the reals or rationals, you can also do Gaussian elimination over systems of Diophantine or Diophantine systems. So linear Diophantine systems where you're solving in the integers. And this problem was particularly interesting to me because one of my PhD advisors was Ravi Kannan, and who actually came up with the fastest known algorithms for solving systems of Diophantine systems. And so it sort of was one of the drivers of my PhD thesis, right? And so the theme of elimination actually then, when I went on to Purdue, became sort of a theme that one of the Purdue elders, Professor Abhyankar, Sriram Abhyankar, really inculcated in all of us as a very powerful way to actually do algebraic geometry and solve geometric problems. And so I became his shishya. I became a student again, took his classes, wrote lecture notes, and he published a book out of, I was one of the scribes, and it has actually been the all-time bestseller in the AMS monographs series, right? Algebraic geometry for scientists and engineers. So really very fortunate to have had such teachers and advisors. And then, all of this sort of led one to think about foundations of deductive logic and logic reasoning, because it was believed, in those days at least, that machine intelligence was going to be actually based on logical reasoning, not not on data, right? The data idea came much later. So, so there were these specialized types of rules where you have only a single consequent, which are called Horn rules, and it turns out that you can solve over Horn rules axiomatic systems defined with Horn rules with a linear time algorithm. So it's extremely fast, and you could build all kinds of systems on top of that. And so a lot of the research was on trying to find, again, special classes of propositional logic formulae on which you could actually get this type of performance that you got with Horn. And so, a series of papers followed. My work in the 80s and 90s was really focused on much of this work. In fact, it was really interesting. I used to come and visit IISc, and also was fortunate to spend some time with Professor G.N. Ramachandran, who was working on these Boolean vector matrix formalisms and was trying to construct actually certain hardware to improve solutions to problems in biology. And I found traces of, what turned out to be the constructs for reversible computing, which again led to quantum computing, were there in his formulations. And so he was already pretty affected by Parkinson's. So we could spend like an hour in the mornings talking before he had to start his medications and so forth, right? That's an unfinished monograph that someday I will work on, because I think there are very deep ideas in there. I actually have all his reports on this work. So anyway, this led to a whole bunch of work at IISc with DRDO at the Center for AI and Robotics, and with a bunch of students at IISc, and we built all these automated theorem proving machines, which of course, for mathematicians in the audience, there was no attempt to replace mathematicians. It was just a term used in this area of computational logic, I think. This was sort of the focus of my work, and as I said, we call this eating sauerkraut with chopsticks. And in fact, that was my fellowship induction lecture in 1997, right? It's called the second half of the Leibniz program. Okay? And so this brings me to 1996. Meanwhile, we had also started discovering that there were interesting things happening with neural networks. And IISc actually had an extremely strong group in pattern recognition and using neural networks. And so we set about solving the world's largest neural network problem at that point, which was on a Hopfield net. And it had to solve about 400,000 synaptic weights using existing computers. It took us 16 days to get to optimality. But these things today seem like toy problems because of how deep learning works with GPUs and so on. But at that point, I think we were the world champions, which was fun, right? And now, I said that Raghavan had a role to play. I think somewhere soon after I had joined, I think Raghavan and Sujata invited me to play some basketball with a couple of students. So this picture is supposed to be Sujata, Raghavan, me, Suri, Venkatachalam, and Gautam Nadig, right? And I think after we, shot some hoops, we were just chatting, and two things happened. I think Suri and Gautam started attending my class in computational geometry because they wanted to know how to compute volumetric properties of macromolecules. And we knew how to do that in computer science. Using three-dimensional Voronoi diagrams, Delaunay triangulations, and various techniques of alpha shapes and so on. So, and in return, they offered to teach me biology, and they turned me on to the protein folding problem. And that's how I got interested in biology. I read the papers of Richards, right? And in Scientific American and so on, and then actually wrote some papers on protein folding on lattices and so forth. But what we started observing in computer science was this remarkable, correlated timeline that was happening in molecular biology and information science, right? Pretty much post World War II, these two were running on the same timeline and with great advances and some crossovers, including the basketball game, right? So this, essentially led us to start thinking about computational biology as a serious enterprise within the computer science department. And so we actually started a lab called the Perceptual Computing Lab, or PERCOLAT, and started doing some work in computational biology. And but we also did a lot of other stuff, right? So I think Professor Rodham Narasimha and M.N. Srinivas at NIAS were all seriously concerned about the digital divide. What would, and Professor V. Rajaraman, right, who's always talking about low-cost computing. So we decided we'll start building a low-cost computer. And we were not overly attached to the PC, right? Or the traditional computer. We thought the time had come to start thinking about mobile computing, mobile devices. And so we started building the computer, right? And then, of course, the computational biology work was going on. So there was an opportunity to get into genomics, and which was the Human Genome Project was on. There was lots of interest in computational tools that biologists needed. And right at that time, Mr. Ratan Tata came as the president of the court of IISc. And I don't know if Raghavan remembers, but he made a statement in the court. He said, why aren't there more startups, right? And that opened the door, because that night we wrote to the director and said, our lab is ready to start up. So what is the process? And Professor Govardhan Mehta, at that time the director, wrote back saying, well, we don't have one, but I'll appoint a committee, right? And so the process started. It took about a year after the committee was appointed to figure out whether this was a worthwhile thing or not. That report was sent to the council, and it was Dr. Raja Ramanna who was the proponent and said, let's do this, right? This had never been done, I think, in India as sanctioned entrepreneurship by faculty, promoting in companies, and with the Indian Institute of Science actually taking a stake in the company as a co-promoter, right? The rules were laid out for that, including all kinds of conflict of interest rules, and there was the Office of Technology Licensing and Entrepreneurship, TOTL, which laid down all these rules, and so as we were called by the press, the audacious Gang of Four, got started. That's Professor Vinay Vishwanathan, Swami Manohar, myself, and Ramesh Hariharan, right? And this is, of course, 25 years back. And, our incubator at that point was inaugurated by Mr. Tata. And so the fun part began, a very different journey after that, on actually trying to build, make some of these technologies translational and have impact, right? As Raghavan mentioned, this required a whole new thought process, and the structuring was actually recommended by an MIT classmate of mine, Dr. Prem Das, who a biochemist who actually had led tech transfer at various institutions, including the Harvard Medical School, Dana-Farber. And so he gave some structure, and then we talked over with Professor Kincha and others at IISc, and this is how it was set up. We actually built our own incubator because there was none at that point, called String Labs, and spun out the companies at String Labs. It was bicycling distance from IISc, so we could go teach our classes and then come to the company. So for about 5 years, we did that, right? We were doing all our academic work and running the companies. And then, in fact, this was such a new experiment that the IIM Bangalore Journal actually wanted us to write up how this whole thing had been structured. So it's been published somewhere in the IMB reviews. But I just want to spend a few minutes talking about the computer, because I think, in a way, it was an extraordinary moment in computing in India, and I think it's also a missed opportunity. In some sense, we were probably at least 4 years ahead of Apple and Samsung in the smartphones era, and we had pretty much all the technologies that have gone into these devices. They made the devices much sleeker, much better marketing, all of that, but the technology is pretty much there. We had built an Android system. We had different names, of course, and it was a touch screen. It was icon driven. It could speak in Indian languages, a text-to-speech system. It was actually, recognized outside India, but in India it was hard to get the funding to really take it all the way, right? But close to 40,000 computers were built and actually used in various applications. And of course, our Minister for Human Resource Development was very happy. This was claimed as one of the top 5 contributions of the NDA government, and we were celebrated in some ways. So it was a great experiment, and as Ravi was mentioning, Sulochana Gadgil actually going to villages and working in climate sciences. I think we were doing that with computing. This photograph here is of children in Chhattisgarh and sharing a computer and learning with computers. We built labs in schools in Narayanpur, deep in the Naxal territories, and in some schools in northern Chhattisgarh. This particular picture made it to the Museum of Modern Art in New York because how technology was getting diffused, right? Was sort of the theme. Sorry, I should go a little quicker. Strand, of course, because of the fact that it was data scientists coming into biology, we kind of focused on building the new tools that were needed by biologists to actually analyze the data that was coming out of better instrumentation that had become available. And it turns out that what they needed was actually machine learning based tools, right? Because in some sense, physical sciences, you can start with formulas and sort of quantitative models, whereas in the life sciences, you often have to work with the data and learn from the data. And in many ways, AI is, or ML is really the most suitable way of doing that. So we built those tools, and actually, it's not very well known that actually these tools built at Strand actually became kind of the gold standard tools that companies like Agilent and Illumina and so on sold worldwide. And actually, if you trace back all the citations, it's actually had a pretty sizable impact in research biology, particularly molecular biology. But then we got excited about the ability to use all of this analytics to enter into this new space of what is being called personalized medicine. In order to do that, we needed to build a whole bunch of tools. I won't go into the details of that, but just to give you a sense of why this was so exciting, I think in 2010, this little boy, Nicholas Volker, is the first patient in the world who is saved because his whole genome sequence is actually analyzed by Howard Jacob, the geneticist. And this story called 1 in a Billion wins the Pulitzer Prize, right? Actually, Ramesh Hariharan and I were in the audience when Howard Jacob presented this case study, and we made up our minds that night that this is what Strand will do as well, right? And so we came back, and meanwhile, important landmark was Angelina Jolie's blog about why she had chosen to do a double mastectomy as a prophylactic surgery to improve her chances of staying alive, right? And of course, later on, she also did an oophorectomy because she was also at high risk for ovarian cancer, right? So these events were starting to build public consciousness as well as start the whole journey of personalized medicine through genomics, and Strand decided to actually play a role, right? So for the first time in South Asia, we had a lab that could actually do next generation sequencing of genomes and provide results, and this was certified by the College of American Pathologists, which is sort of the certifying authority for clinical diagnostic tests. And Dimple Bawa, for example, is the Angelina Jolie of India, right? So she's also a person who's come out and spoken about why breast and ovarian cancer and women's health needs much more attention. And similarly, thousands of other patients have been so generous and willing to share their stories. And today, I think we do over 1,000 cases a month, right, of this type of diagnosis for clinical work. So Strand's evolution essentially I'm sorry for the dollar figures, but that's the kind of money one has to raise to build, these tech companies. Starts out in informatics, then goes to the diagnostics, and then the clinical impact starts showing a few years later. And then you get to a point where you have to know how to scale this. How do you actually take this to population scale? You have now proved that the technology works, that it's actually useful, but somebody then has to commercialize it and take it to scale. And a couple of professors like Ramesh Hariharan and I are not, the best equipped to think about that kind of scale. So fortunately for us, two things were happening. One was that the cost of sequencing was coming down extremely rapidly, right? If this is obviously a log scale on the y-axis, so the straight line there represents Moore's law, right? Which is, of course, the much celebrated law of computing doubling every 18 months, right? In its capacity and the cost coming down every 18 months, halving every 18 months. If you look at the cost of genome sequencing, it sort of stays with Moore's law, and then something called next-gen sequencing takes effect, and it starts coming down much more rapidly, right? So it's actually the tipping point that everyone talks about is when you can sequence 1 gigabase of nucleic material for less than 100 rupees, right? Where are we today? We're at about 200 rupees. If you can do it at volume, if you can use the best machines, that's where technology is today. So this technology is helping us. Eventually, it'll help us much more if somebody could actually build sequencers in India and actually make all the reagents that we need in India. Instead, like China has done, right? We haven't done that in India yet. And when you're able to reach that tipping point, commercial engines like the Reliance engine is willing to step forward, right? So that's basically what has happened. Mr. Ambani has now acquired most of Strand. Ramesh and I are still part of it, but he bought out all the investors and everyone who is not working for the company, and is basically now wants to democratize genomics, in the sense that take it to population scale, right? So that's the journey now for Ramesh. I've retired, so I just sit on the board and so forth. But what was also exciting was what was happening in synthetic biology. So if you think sequencing and reading the genome is a well-solved problem, now the next problem is how do you actually manipulate genomes and how do you take it to that next level, right? And this was actually a challenge thrown out by Eric Lander, saying we must learn to program cells with the same facility with which we program computers. And this was around the 10th anniversary of the Human Genome Project. And quite interestingly, the top part is more about bioeconomics, pointing out why governments should invest more in some technologies which actually make a difference. And that was Obama's State of the Union address when he said, investing $5.6 billion in the Human Genome Project and continuing work on genomics actually yielded close to $800 billion to the economy, right, by 2011. And therefore, he went to Congress and the US Congress and asked for more funds to sequence 1 million US citizens, which is called the All of Us program. And making arguments like this is important, and we've not done that, right, as a scientific community, to actually talk about how impact, and this could be one of the things that was suggested that the academy can do, right, and to convince policymakers that there are certain technologies that are very critical and have to be supported right from the foundational level to full translation. So, but the neat thing was Jennifer Doudna and Emmanuel Charpentier, just soon after Eric Lander's article comes out, they publish the CRISPR paper saying, there's this technique now for doing cut and paste operations in genomes, and it actually comes from bacteria, right, protecting themselves against viruses, right. So there is a mechanism that is used in nature, and how that can be converted into a technology, right, and which has much wider purpose. And this is all Kevin Kelly's stuff coming to life, right? Your engineering biology and your and you're learning from human engineered systems, and of course now there's a whole set of implications of this. Primarily, people have thought about this from a healthcare point of view, but in the future we're going to see this use of CRISPR in engineering, in bioindustrial products, in cleaning the environment. I think our best friends are going to be the microbes, right? And if we can actually edit those microbes to clean the environment, to convert Napier grass into compressed biogas, or do leaching of rare earths from waste, without acid and burning and so on, which is how it's done today. I think all of this can come from engineered biology, and I think that's the power of this stuff. And just for maybe some of the students here, by the way, I'll just go back. Jennifer Doudna wrote this book called A Crack in Creation. Don't read The Code Maker by Walter Isaacson. This is the book to read, right, about CRISPR. Walter Isaacson makes people larger than life. I think this is the real discussion, written by Jennifer and her student Sam Sternberg. So we've had good access to Sam, so it's been a lot of fun working with Sternberg in CRISPR. Professor Sternberg, now at Columbia, shared with us this video of an electron microscope actually visualizing the cis cleavage done by Cas on, as you can see, the separation happens. Now, sometimes it happens in 30 seconds, sometimes it takes 90 seconds, sometimes it takes, 20 seconds. So, one of the ways in which you can actually understand this is to do quantum simulation, right? And ORNL, the Oak Ridge National Labs now, have a quantum simulation set up to actually look at the cleavage efficiencies intracellular, right? So it's interesting how different sciences come together. The trans cleavage is now the CRISPR guide has found a match in the DNA or in the new RNA in the and once it has found the match, this this Cas enzyme will cleave any nucleic material in the vicinity. So if you throw in reporters with fluorescent tags, you'll actually get a fluorescence image. And you will know that the CRISPR guide has actually attached, right? And so you can, this becomes a diagnostic. Right? And so as you know, during the pandemic, India tried, I think we tried to build something called Feluda, which the Tatas then tried to manufacture and take forward, but it didn't quite succeed. It was, the technology is so fast moving that, you know, I think it's getting better and better. So now seemed to be a good time to try this. So the mission my group at CRISPR Bits is now working on is to actually develop eventually a chip in which you can do multiplexed CRISPR-based detection of pathogens, of antimicrobial resistance markers, you know, any type of nucleic pattern recognition that that could be useful and the whole idea is to do this at a point of care. In fact, we call it point of need testing, because CRISPR is so sensitive and doesn't actually need thermocycling, so you don't even need a PCR for amplification. You can do this with isothermal amplification, like RPA or LAMP, and this can be done very effectively at the point of care. Building such a chip is quite a process, and that's what we're embarked on. But we've decided that we will name the chip the VV chip in memory of Professor V. Venkatraman from the physics department, the late Professor Venkatraman, because this was, I mean, he was always interested in this idea of building a lab on a chip. So that's, this is the project we're on. I'm quite interested in also various aspects of health, and with Professor Ravi Nanjundaya, we are now looking at climate and health, and we're going to actually be looking at how heat stress impacts health. As we start, you know, I think no one is really quite understands what climate change and, you know, the changes in particularly the heat waves and that impact it's going to have. So we've just started on a study, and you'll hear a lot more about it tomorrow from Professor Nanjundaya. Just want to end on this slide, which basically talks about the journey that in some sense I joined, well, technically in around the early mid-70s when I was at BITS Pilani, and you know, Professor Ram Gopal Rao is here, our Vice Chancellor. So, you know, I think that was when I got interested in intelligent control systems and electrical engineering, and that journey has gone through here. I'm now 72 years old. I'm not sure there's a lot more I can do, but this is the last project that I really hope that we will see some impact on, which is how do we get to universal healthcare. And at least on the diagnostic side, I think the journey that been on could possibly help, right? So I'll end there. Thank you very much, and yeah.
Yeah, that was a very fascinating talk. Thank you so much, Vijay. And it's actually interesting that from a basketball game, Vijay started so many companies, and as it turns out, the other players there, Gautam and Suri, started Minvax, not Minvax,
Metahelix,
Metahelix, which is a very successful agri-biotech company, which was sold to the Tatas in 2016. And then Gautam and I started Minvax in 2017. So this, any questions?
You mentioned you are 72 years old.
Yes.
Actually, you are 72 years young. Because these days people talk about super aging.
Right.
Super ages. So
Yeah.
we'll see you in the 100th birthday.
No, thank you so much. I am actually living my second life. I was in the wrong place at the wrong time, took 4 bullets from a terrorist, and this was exactly 20 years ago. And so I'm just very grateful that I've had those 20 years. But yeah, thank you. I certainly hope to keep working as long as I can. But it's
very fascinating talk indeed.
Sure,
I was
you.
just wondering, as you mentioned synthetic food, whether that refers to precision fermentation.
Yes, absolutely. So I'm sorry, let me put that back. So synthetic food, precision nutrition, and so forth. In fact, there's several areas that we are working on in precision fermentation, including, of course, production of various biologics and so on, where, again, microbial strain optimization, strain engineering is the production of biofuels. But in food, also working very hard on food safety. It turns out that, something like 6.2 million people suffer from food poisoning a year, and over 4 lakh people actually lose their lives due to poisoned food, right? And our regulatory infrastructure for testing food safety is extremely poor, right? And we still rely on cell cultures, and that's so tedious, and the requirements are also very low, right? So from that perspective, we're hoping that this CRISPR type of testing can actually help, test early so that if there is contamination, it can be.
So that PHYX in Bangalore, do you have anything to do with it? There's this company called PHYX in Bangalore.
No,
And they produce a lot of milk products, casein and whey and other things.
Right. No, I don't have any connection. Yeah, but it's such a huge industry, the food products industry, that it's taking off, and there's all this wellness and longevity, and I think as the comment was just made, there's a lot of focus these days on wellness and nutrition. But food safety is a concern. I think actually manufacturing of certain plant-based foods which have a protein value and so on, all that is starting to happen. We're also looking at how to grow shrimps or seafood from cell-based methods. There's lots of engineered biology that can happen. Right. Please, yeah.
Wonderful lecture, of course.
Thank you.
In biology, nothing makes sense unless you see in the light of evolution.
Right.
That was said by Dobzhansky.
Right.
Now, the CRISPR technology is respected, of course.
Right.
But how can one say that we can control the evolution? Evolution is not dependent on our whim and fancy.
Right.
It is something you cannot control. That's
Sure.
point number 1.
Yeah.
Point number 2, your presentation is absolutely fascinating. How do you think in a population, in a country like India, 147 crores of people, that the diagnosis at the sequence level can be made available for common people. Even ordinary women in the society do not have enough money to go for mammography.
Correct.
Let alone talking about BRCA1, BRCA2, and so on and so forth.
Sure.
Of course, Angelina is a different story. She's a very rich lady, of course. So maybe that you want to little bit make some comment?
Sure, sure.
Of course, I have so many questions, but I'll remain
Right.
civilized today. Thank you so much. Yeah.
Right.
Go on.
So it's so the answer is that genomics by itself is not the answer, of course, right? There are various types of imaging technologies which could be used at an initial screening level. I think where you want to go to genomics is for confirmation. Right? Try to use more affordable screening methods to start with, and then when you need the confirmation, you go to a molecular test. So that's part of the...
Just one thing. I think, for the students at the back, this is an opportunity that you should take advantage of.
Right.
Professor Vijay Chandru won't be around, and he'll be here today and tomorrow. So you should take the opportunity to talk to him. If you have any questions, please, there's no such thing as a stupid question. So feel free to raise them.
Right. And, I'd be happy to chat after as well, if anybody wants.
So Vijay, except for those 4 bullets, what has been the biggest challenge, scientific challenge in your journey?
Oh, in which journey? Right. The academic
that you described to engineered biology, from
Right.
computing to engineered biology.
Sure. Well, some of the early challenges were the usual academic challenges, right? Tenure and getting elected to the academy and so forth. But that apart, I think everybody thought this was really strange that someone who is an academician would want to go out and start companies and stuff like that. But I've always maintained that if you are an active researcher, an academic researcher, then you have all the makings, right? You know how to, advertise your work, right? You know how to make presentations and raise money, right? You know how to run a team, right? You have all the tools you need. You have to work at a different speed. You have to work at a different intensity, right? When the phone rings, it's an opportunity. Don't let it go to voicemail, right? There are some small changes that you have to make, but otherwise you have the basic, training to be an entrepreneur. I see, the thing was also role models, right? When I was at MIT, I would see mathematicians like Dan Kleitman, or Giancarlo Rota, who were, very, very classical mathematicians, but, consulting, working on, real problems on electrical networks, on telecom networks, and using graph theory. And so I think it's just a mindset, right? If we're willing to change that mindset, it'll come naturally. But the other problem is, of course, the challenge of actually finding patient capital that is willing to support deep science or deep technology that may not give returns very easily. That still remains a challenge. And I think the government has done a fantastic job at the startup stage, right? Today, any young person who wants to be an entrepreneur can get that seed funds. If they have a reasonable plan, reasonable idea, they want to take it forward, there is 50 lakhs or some kind of a startup grant that you have. How you are able to go from that idea to a real proof of concept and take it close enough to a product that would actually make an impact, there is still a problem of finding the capital for that. And I think the trend is there. You're starting to see early stage investors and so on coming in. But personally, as the challenge was, I think, it's a long journey, and how to keep your emotional IQ going is hard. And I think the lesson, at least from our journey, is that if you're not alone and that you're a gang of four, that it helps a lot. Right? Because I think it would have been really difficult to do this on one's own. Right? There are people who do, so it's possible, but I think the reason we were able to sustain for all these years is that we can talk and talk through problems. Thanks.
Some basic query. We have evidence in plant genomes that respond differentially when the temperature goes up.
Right.
And one of the evidence shows that, at least to my knowledge, that as the genomic shocks occur during high temperature or elevated locations, the retrotransposons, which are fundamentally major part of plant genomes, they become activated and they start inducing the epigenetic changes,
Right.
and leading to perturbations in the transcription and translation. So, is it one of the technologies should be aimed at somehow reducing this epigenetic load, which is not directly mutations, but some of them will go for imprinting type, a second generation effect also. So this is a concern for geneticists all along.
Right.
And those of us working on transposons for a long time, we know that maize genome, for example, is 75% is nothing to do with maize gene, maize DNA. It's all retroviral, retrotransposon origin. And so is millets. Barley is 85% of DNA is nothing to do with barley. Okay. It's coming from retrotransposon, retroviral sequences. So this technology that we have developed will have a tremendous effect if we have somehow target the epigenetic changes and keep the transposon silent. At higher elevations, transposons get activated.
Yeah.
In retroviral sequences.
I must admit we are at a very early stage,
Okay.
right? But so we are open to working on applications.
Okay.
If there is something that you feel has potential.
It has tremendous diseases, many diseases,
Sure.
which are reversible, are associated with epigenetic changes posed and induced by these transposable elements, which can jump into chromosome just like that.
Right. No,
Yeah.
Absolutely.
Okay.
Thank you.
So thank you, sir, for the fantastic talk. I have a question which might be a basic question, but I want to ask, like, how can we be sure that a complex AI models of like protein folding are like not wrong? Because like AI models can be wrong. So how can we be sure that what we are modeling is correct?
So you're talking about hallucinations by AI models, or those types of rogue sort of behavior. Absolutely, I think there's a big challenge, and that's why I think anything generated from AI will have to be taken as a lead and then tested, appropriately. Anything to do with biology, you'd have to go to the lab, right? And it's not just AI-generated lead molecules being, thinking that itself is going to lead to a new drug is sort of, still a fantasy. It's not even a group like Isomorphic Labs built by DeepMind, or David Baker's lab at University of Washington that does the Rosetta fold, which is like the other version. There's AlphaFold and Rosetta fold. And none of them are actually claiming that they can just generate this through AI and it'll work. There is, I think the more dangerous problem is with LLMs and just general these GPTs and taking what they say very literally. The bigger problem is the following, right? They have already ingested almost all the data that's available publicly. Right? From now onwards, they will be generating synthetic data and using that to keep learning. And you can imagine how biases will perpetrate. And so how you control the quality of what comes out of these AIs will become more and more of an issue. And I think you're absolutely right. AI has to be taken with a pinch of salt. And I think the more than a pinch. I think you do have to also, we have to be careful about building good curated data sets for training. And Raghavan and I were talking about this. Just because somebody says they have 100,000 images from X-rays doesn't mean you can actually build an AI on top of that, right? Those X-rays would have to be annotated, they'd have to be curated, you'd have to add the phenotypic information associated with each of those X-rays, and then you can learn something from that. Now, as I guess not just India, but many countries have had problems with actually being able to create these high quality data sets and use them to build AIs. But that's a key step. And one of the projects at IISc now is called MIDAS, which is on gold standard data sets, which is funded by ICMR to actually create these gold standard data sets for being able to calibrate AI methodologies on them. So, yeah. So
Thank you, sir.
It's the usual adage, garbage in, garbage out, right? So if the data is no good, then your AI is no good, right? So.
Thank you again.
Yeah.
What intrigued me most was that you began with the Leibniz program. That there should be a language which could be written in a symbolic form. And of course, later on, a lot of it materialized in the way Boolean algebra and then French detail. Right. So the question I have is that, and then you brought out some corollaries from that in those symbolic logic, and it's several corollaries. And listening to your fascinating journey, I was wondering whether all this was hidden in what those formulations had. And if you were to recommend a program for a creative journey like yours, would you say one should begin there? And I had a second question,
Right.
and that was whether, that's just information, whether in the AI that you use, you do put any tests for causalities.
All right. So just two quick responses to that. One is, as far as training is concerned, and I think what was most valuable for me was physics, actually, because in those days, an electrical engineer, as an undergrad, did 5 years and did 8 semesters of physics. Right? And that training in physics basically helped one think, be able to move into very interdisciplinary areas, into many disciplines, because in some sense, physics teaches you to model, right? And to build mathematical models of any sort of abstract problem you encounter. And I think that, so if anyone is choosing an undergraduate discipline, don't leave out the physics. It would be my, at least it helped me, right? I'm sorry, the second part was? About the causality. Oh, causality, yeah. Causality is a tough problem. Judea Pearl, for example, has worked on causality. He was one of my teachers at UCLA. Even in things like LLMs and so on, when we actually we had to build natural language processing engines to read, because in those days there was no deep learning at Strand. In 2004, when we actually read all of PubMed, Medline, we used an NLP engine to read. And we actually programmed whole sentence parsing. So we programmed the English grammar into automata, and we actually could read and gave it the full lexicon of biology so that it could actually read these papers and understand and build the graphs, the underlying knowledge graphs, which then could be used for interpretation. Now there, A regulated by B is very different from saying A and B are related, right? So you actually had to use the rules of grammar to actually understand that what this paper was talking about was A regulates B, and actually draw the arrow the right way, right? So in some sense, we did work on causality and using natural language processing, but it the broader problem of causality from data is a hard AI problem. It's still not been cracked, right? So.
Okay. Yeah.
So when you came in the field of research, so as a student, what was your perspective towards the future?
Actually, I was very excited to play cricket. I'm just joking. I think where I got really excited about research was Pilani was kind of an interesting place. But Pilani, we would get interesting visitors to come and speak to us, and why am I forgetting his name? There was an Englishman who was one of the founders of cybernetics in the UK, and he was a friend of the director then, Professor C.R. Mitra, and he came to spend a semester at BITS Pilani, and he gave us lectures on cybernetics. And I think that excited me. And cybernetics and intelligent control was where AI really came from, right? In fact, at MIT, long before the AI lab was set up, intelligent control and cybernetics was the field invented by Norbert Wiener, and the cybernetics lab was where I did my PhD work. So yeah, I think just having that exposure to an advanced researcher coming and giving, he just gave a few lectures in the auditorium, and we got to listen, and that excited me. And I spoke to the director after that, and I said, I want to work in this field, and he suggested places that I could go and study. Right. So.
Thank you, sir.
Sure.
Oh, okay, I think we'll stop here. So thanks a lot,
Sure.
Vijay, for this fascinating journey, and in building companies that are not software or logistics, but making real things, and long and very difficult. journey. Thanks for sharing it with us.
Thank you.
So it's been a real pleasure to have you.
Thank you.
Thank you.